I work on a disease that affects only 14 people in 1 million. Have you ever heard of PIK3-related overgrowth syndromes? Until 2 years ago, I had never heard of them, and the reason is because they encompass a group of very rare and difficult to classify diseases. Patients affected by this disease present abnormal growth of various body parts or defect in the vascular system, and this is due to changes known as mutation in the PIK3CA gene. This gene is involved in making a very important protein that is actually fundamental for the cells and help regulate their growth, their division, and their survival. You can imagine how important it is for the cells having this protein working properly, and the problem arising when it's not well regulated. Since PIK3CA mutations are not inherited but originate during the embryonic development, they are not present in all the cells of the body. Imagine the cells that compose the early embryo as the tiles that compose a colorful mosaic, with each of them having a different color. Now imagine that at a certain point, one of them acquires a mutation that makes it lose its color. All the cells deriving by this one will be white. In the same way, only the cells and consequently the tissue deriving by this one will carry the PIK3CA mutation. Depending on the time in which the mutation occurs and the cell type, different tissue may be involved, individually or in combination, and have different clinical consequences. We in the lab noticed that cells affected by this PIK3CA mutation show a different way of getting nutrients in comparison to their healthy sisters. In fact, they have a predilection for glucose that distinguishes them from the others. So now we have two objectives. The first one is to explain how this change happened in the first place. And the second one is to exploit this particular characteristic of the mutated cells to target them with a specific drug. In the lab, we have already found a drug to treat the patient, which has now become the gold standard for the treatment. But there is still room for improvement. What we are planning to do now is to combine the drug with other treatments specifically directed to the mutated cells. Some of you may be wondering, how will it be possible to target treatment only against mutated cells? Well, probably thanks to their specific predilection for glucose. The idea is to try to make glucose less available or more difficult to use for these cells, and thus to try to make them more susceptible to the therapy. Doing this, we hope to develop a target therapy and improving the quality of life of those patients that are less responsive to our gold standard treatment. Thank you.